Welcome to this lesson on prophase, the first and longest stage of mitosis. Prophase is when the cell begins preparing to divide, and the most important event during this stage is chromosome condensation. This is when the genetic material becomes visible under a microscope for the first time. Let's explore what happens during this critical phase of cell division. Prophase is defined as the first stage of mitosis, where chromatin condenses into visible chromosomes. Before prophase begins, the DNA exists as loose, thread-like strands called chromatin that are scattered throughout the nucleus. During prophase, these chromatin fibers begin to coil and fold tightly, transforming into the distinct chromosome structures that can be observed under a light microscope. 4 major events occur during prophase. First, chromatin condenses into compact chromosomes, making the genetic material easier to organize and separate. Second, the nuclear envelope, which is the membrane surrounding the nucleus, begins to break down. Third, centrioles, which are small organelles, move to opposite poles or ends of the cell. Fourth, spindle fibers start forming between these centrioles, creating a structure that will later pull the chromosomes apart. Let's take a closer look at chromatin condensation. Before prophase, chromatin exists as loose, thread-like fibers scattered randomly throughout the nucleus. You cannot see individual chromosomes at this stage. After condensation occurs during prophase, the chromatin transforms into tightly coiled, visible structures shaped like the letter X. These are the chromosomes, and they are now organized and ready to be separated into daughter cells. Each chromosome that becomes visible during prophase has a specific structure. It consists of 2 sister chromatids, which are identical copies of the DNA. These sister chromatids are joined together at a region called the centromere, which acts like a connection point holding them together. It's important to understand that each chromatid contains identical genetic information because the DNA was replicated during the S phase of interphase, before mitosis began. During prophase, the cell constructs the spindle apparatus, which is essential for separating chromosomes. Centrioles migrate to opposite poles of the cell, positioning themselves at each end. From each centriole, spindle fibers begin extending toward the center of the cell. These spindle fibers are made of protein structures called microtubules, which are long, hollow tubes that provide the mechanical force needed to move chromosomes during later stages of mitosis. Another critical event in prophase is the breakdown of the nuclear envelope. The nuclear membrane, which normally surrounds and protects the nucleus, begins to disintegrate during prophase. This breakdown is necessary because it allows the spindle fibers to access the chromosomes inside the nucleus. By the end of prophase, the nuclear envelope is completely gone, and the chromosomes are free-floating in the cell, ready to be attached to spindle fibers. To summarize prophase, 4 key events occur during this first stage of mitosis. Chromatin condenses into visible chromosomes, making the genetic material organized and ready for division. Sister chromatids remain joined at the centromere, ensuring that each chromosome consists of 2 identical DNA copies. The nuclear envelope breaks down completely, removing the barrier between chromosomes and spindle fibers. Finally, the spindle apparatus forms, with centrioles at opposite poles and microtubules extending between them. Once prophase is complete, the cell is ready to move into the next stage of mitosis called metaphase.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.